was working, maybe we don't need to record this, or maybe we will cut. So he was working also on a development of improvement of the atomic bomb.
Yeah.
What?
In addition, he was one of the
Was an investigator.
but also reservoirs in Brno. So together you helped them to establish reservoirs in Brno. That's one of the, how many of us? Ranka, Maro? Yeah, how many? Chuka. Yeah, you visited reservoirs. It's one of the most important places on planet regarding ecotoxicology. In addition, Professor Giesy was involved in the COVID-19 crisis. So amazing life, amazing achievements, amazing everything.
Do we have time for a quick story?
Yeah,
Yes.
please.
It's funny how the world works. Funny things happen. So I used to live in Germany. German's my first language. So then French, then English. My professor, Otto Hutzinger, was very famous for dioxin analysis. And people were always coming to our laboratory, and one guy from Czech Republic came, Ivan Holibek, came, and Professor Hutzinger said to me, he says, he's your problem, you take care of him. He's a Czech guy, he doesn't speak any German, you speak English, you speak German, you take care of him. So I was taking care of this guy, and we became really good friends. He's a really nice guy, really smart guy, and he invited me to the Czech Republic. So I went there in the 1970s sometime, and he was so proud of his laboratory. He took me, it was a little room with no heating in it, in an old warehouse, and he had GC with an FID. That was it. That's all he had. And he was so proud of it. And he wanted to show me, and I was like, ugh. Because, I was working in Bayreuth, we were using magnetic sector, high resolution mass spectrometry, and I was like, ugh, okay. Anyway, I really enjoyed those people, but they had nothing, absolutely nothing. And he basically said, I want you to meet somebody. So we had a dinner, and I met one of his students, Jana Kanova, and also one of his undergrads, Clara Hilscherhoff. And he said to me, he said, Clara is a very, very good student. I want her to get a better education than she can get here. I want you to take her. So I did. I took Clara to my lab, and she got a bachelor's, she got a master's degree, then a PhD. She came back 2 or 3 times as a PhD. Ludek Blaha also was postdoc with me. So many of those people, and then they asked me to be the chair of their external advisory panel. I've been with them through now 4 new buildings. They keep growing, getting better, new equipment, 4 new buildings, unbelievable. And I love them. They're great people, they're great scientists, and from their former times, now that they've been released to grow and be free and independent, they just blossom. So it's one of the pleasures of my life to watch them. But it came from this weird thing, like you take care of this Czech guy because he doesn't speak any German. Very strange. But it worked out really, really well. Okay, what are we doing now? Oh my goodness.
And by the way, we are talking about Otto Hutzinger. The dioxin meeting is very important meeting about the dioxin sulfurous. Now these days mainly about the perfluorinated compounds. And if you're really, really good, you will receive an award at the dioxin meeting, and the name of the award is Otto Hutzinger. And Ivan Holobek unfortunately passed away last year, head of the Resotox, but now Jana Klanova is
Yeah.
What place in Czech Republic was it?
Brno.
Brno.
Oh, okay. I did my master in Brno when I was young.
Really?
But in BUT, so Brno University of Technology. I don't know if it's connected.
It's at Maastricht University.
Maastricht, so it's a sister university.
And then they also gave me honorary PhD from Maastricht University. So we're like alumni.
Yeah.
Alumni.
No, and you know, it's been really interesting to me over whatever, 40 years, 45 years, going to Brno all the time, watching the changes. You know, when I first went there, there was a lot of like damage from World War II, trees growing out of buildings and stuff. Now, beautiful city, very nice. Okay, how much time do we have?
1.5
Hours? Perfect. All right, going to take off. Okay, I want to talk about environmental forensics, what it is and what it can do. Anyway, I've been lots of places, and lots of people have paid a lot of money for all the research that we do. I was a professor at Hong Kong University for maybe 20 years. That's my lab in Canada. So they built this building just for me when they brought me there 20-some years ago. We do research all over the world, global research. This is in Antarctica, this is in Michigan. I work a lot with birds. Many of my former students and postdocs are now very influential in the world. I'm real proud of all of them. This is in Korea, this is in China. This is Miguel Mora, one of my former postdocs. Now all my students and postdocs are retiring, so anyway, one of the keys for what I'll say today is I could never decide if I liked chemistry or biology better. And I was always being pushed one way or the other, and professors were pulling me. I felt like a piece of cheese. And finally I said, I'm not going to do that, I'm always going to do both. So we do work with humans, we do human risk assessment, measurement of residues, toxicology in humans and wildlife. I do laboratory and field. We do basic science and applied. Sometimes we have to develop things to do the science we want to do. And I do everything from molecular to ecosystem. So I'm nuts. And that's the big thing, is that I'm completely crazy. And we have what I call a student-based research program. I'm now retired from the University of Saskatchewan. I still have my home in Saskatoon. But now I'm a professor at Baylor University in Texas. And some of you know my colleague George Cobb was at the meeting. Jan Lee, another one of my colleagues, was at the meeting. So I think we have one of the premier programs in North America, if not the world. And we cover all the bases, everything from policy to mass spec and toxicology, environmental science. So, what is environmental forensics? You know the show CSI? Anybody? Anybody not know the show CSI? Okay, so crime scene investigation, CSI. Well, we look for novel contaminants in the environment, and we try to do risk assessments to determine, are they a problem, are they not a problem? And it's a lot like CSI, except without the body, mostly. Sometimes the body, in our case, is wildlife. It's birds, and the question is, what killed the bird? There was a pigeon outside my hotel today that was dead. I should grab it and find out who killed it. And so on the CSI show, remember, they were always sending things to the mass spec. And it was always amazing to me, you know, that immediately they'd say, oh yeah, well, you know, it was an oil that was only manufactured between 1992 and 94 in this place in Louisiana. And I'm like, no, that's not how it works. And they're always sending it off for what? DNA analysis. So today I'm going to talk about mass spectrometry, different than what we talked about earlier, and some DNA analysis. Just going to skip some of that stuff. Skip that too. Okay, so we use these different technologies to answer questions about the environment. And we use top-down and bottom-up approaches. We use inductive and deductive reasoning. So we make observations, and then we develop hypotheses, and then we test those hypotheses. And based on what we learn, then we can make other hypotheses. So we're always doing inductive, deductive reasoning. Not one, not the other. They both have their place, they're both useful. And I have arguments with professors that say it should be one or it should be the other. But we use both. And so we use these top-down and bottom-up approaches. I'll show you some bioassay type things we do. That's sort of at the molecular level, and we try to predict what will happen. Other ones, we look at what's happening at the ecosystem level and try to figure out maybe what's causing that and work down. So that's bottom-up, top-down. And we try to come up with causes. We do a lot with molecular mode of action, try to figure out what's causing certain effects. And mode of action is really important because it tells you what chemicals or environmental stresses need to be considered together. Okay, so if things have a similar mode of action, we might be able to group them together and probably need to group them together to assess the overall risk. If they're independent, then we can maybe do them independently, separately. So that's important to know. So we work a lot on pathways and mechanisms of effects based on adverse outcome pathways. So some of the chemical issues, and I'm not going to talk as much, I think, as I plan to because we spent so much time this morning. But we use untargeted identification of chemicals with ultra-high resolution mass spectrometry. So I've identified a lot of chemicals in the environment, many for the first time, using this approach, an untargeted approach. We look and see what we find, but we haven't said what we're looking for to start with. I was talking to a few of you, it's not easy. Okay, people talk to me and say, well, it's easy for you. It's not easy. We're going to talk a little bit about bioassays and how they work in the whole process. And one of the things we do a lot of is a toxicity mechanism-based tox balance. So we predict all the chemicals, like we use this for dioxins and dibenzofurans. So I've developed a genetically modified cell line that responds to dioxins in proportional to their potency. And it's a transgenic cell line where I've put luciferase, which makes light in the fireflies. And so every hit on the receptor proportional to the strength of the effect, I get a flash of light. So that means I can integrate the effects of all of the dioxins and dibenzofurans and all of the other chemicals that act through the aromatic hydrocarbon receptor together. And then I can measure the chemicals and have relative potency values for all of them and add them up and determine have I identified everything that's causing that effect or not. So it's a potency balance, just like we were doing with the mass balance. But we use bioassays. We do use something called effect-directed fractionation and identification. And I wish I would have had longer to show you some more examples of some of these things. But what's a good example? I was asked to work with the Southern Nevada Water Authority because they thought they had a problem of endocrine disruptors in their drinking water. And so we have another bioassay that I've developed that measures endocrine disrupting effects. And we were able to use that. And interestingly enough, here in Europe, people had seen a similar effect, and they had determined that it was a chemical called nonylphenol, which is in detergents, and they were banning it because they thought it was an endocrine disruptor. It was causing estrogenic effects. Through the bioassay-directed fractionation and identification. What we do is we fractionate our extract, run it in the bioassay, we determine where the activity is, and then I analyze that. And what we found out was this estrogenic effect for this chemical that was being banned wasn't due to nonylphenol. It was in fact due to endogenous estrogen from people. And not only women have estrogen, men have estrogen too, that they were excreting, but it was also ethynyl estradiol, which is a birth control. It was much more potent. And so we identified it was that, not nonylphenol. So if they had, well, they did ban nonylphenol, it wasn't going to take care of the problem. So anyway, that's bioassay directly fractionation and identification. Today I'll tell you about pull-down assays. One of the questions we would have is chemicals in the environment, what effect they might have on the body. So I can take chemicals or an extract and then see what proteins it binds to or what molecular pathways are up or down regulated. And in that way I can figure out what effect those chemicals might have. I can go the other way with a known chemical and ask what effect it will have. So I can do it really kind of both ways. And we use these mass balance approaches. I don't know if I'm going to spend any time on this. I talked to you about it this morning. But this is just an example of what we can do with these sort of mass balance approaches. So this is with brominated compounds. And what you're looking at here are some different animals. And these are the proportions of the different brominated compounds. And then I did this mass balance approach where we looked at how much organically bound bromine there was. And what we found was that about 1% of the organically bound bromine could be explained by these chemicals. 1%. That meant 99% of the organically bound bromine wasn't brominated flame retardants or anything else. And then we go about trying to figure out what that other 99% might be. So, and through here, again, these are all available. I'll leave all this with you. You can go back to these individual papers for the details. There's our mass spec. I showed you that earlier. We've already covered this morning. Sorry. So I can skip all of that because we know how we did that. We've done this in sediments, and this is from the Great Lakes. I was running a big program between the United States and Canada. I'm a dual citizen, so I can work both sides of the border, no trouble, and was working for the US EPA doing this. And we were doing sediment cores for all over the whole Great Lakes. We found, in this case, we were looking for brominated compounds, and we found 2,520 peaks that had never been identified before. And so a lot of the work we were doing was to identify what those chemicals are, and then to determine what toxic effects they might have, what their mode of action is. It's again how we figured out all the structures for all these. There's our library I told you about this morning. And I told you I wrote a program. There's an R package available to do all this. Some of the ongoing studies. So a lot of things going on with brominated compounds. I'm going to spend a little bit more time on this because this is a real world problem. I think you have this problem here in Serbia. Eutrophication is a problem. So let's spend a little bit more time on that. But we're going to use our diptych frag untargeted screening that you now know all about on a real world problem. So, real quickly in disinfection, in North America, for drinking water, we still use a lot of chlorination because it's cheap, it's effective, and it just plain works. But one of the problems is it forms disinfection byproducts. So let's talk about what those are and how this happens. So disinfection byproducts, or DBPs. So in surface water, natural organic matter, things like humic and fulvic acids, proteins, carbohydrates, a number of things that are in water that give it that sort of yellow color or brown, can be chlorinated. So if you chlorinate drink water for disinfection, all of those chemicals can be oxidized by chlorine, which is a very strong oxidizing agent, and form chlorinated byproducts. So we know about this, and the government regulates some of them. These are the only ones the government regulates. So the trihalomethanes, and the total of them cannot exceed 0.1 milligrams per liter in drinking water. And haloacetic acids, and the total can't exceed 0.08 milligrams per liter, 80 parts per billion. Now we know that a number of these chemicals, chlorinated, and you'll see here there are also brominated compounds, can cause cancer. There was a study done in the United States. It turned out people in New Orleans had much higher incidence of esophageal, stomach, and intestinal cancer than the rest of the United States. Does anybody know where Los Angeles or New Orleans is?
Yes, southern state.
Where is it? What goes through the middle of it?
South Mexico.
So Mississippi, all right? Mississippi River. What is the Mississippi River? It's our Danube, right? It's a major river and it runs through the whole United States. And because the people there had such high incidence of cancer, the thinking was it was something in the drinking water coming down the Mississippi, and they were concerned about pesticides. That was the big concern. So I was working, doing what I do, and they contacted me and said, would you work on this? So I led a whole team to look at it. Long story short, we found out it was the drinking water, but it wasn't pesticides coming down the river. It was disinfection byproducts that they were making when they chlorinated the water. Okay, so this was a discovery 30 years ago. When we started to look at this, I found 600 unidentified chemicals in our drinking water now that are chlorinated and brominated and chlorobromo compounds. 600. And the government was monitoring only 9. And so when I reported this, people got very upset. So what's going on? And a lot of these, and the most potent ones, and the ones that were occurring at the greatest concentration, were not chlorinated, but brominated or chlorobromo. And so they weren't on the radar screen for the US EPA. They weren't monitoring for them. So we applied our technology to look at drinking water. So here's where I live now. I live on the high plains, the prairie, in Canada. We're 500 kilometers north of the US border. We're in the rain shadow of the mountains. We don't have a lot of groundwater. All of our water comes from snow melt in the Rocky Mountains, down the north of Saskatchewan River. And this is a side channel called Buffalo Pound. And that's what the water looks like. Okay? So when I arrived here, one of the issues they wanted me to work on was this pollution problem. What was going on and how to solve it. So we don't have a lot of groundwater. All of our water comes from the South Saskatchewan River, comes from the mountains, flows through a number of metropolitan areas before it gets to us, which means a lot of those wastewater treatment plants have minimal treatment for nutrients, and so phosphorus builds up. And so that Lake Diefenbaker, which is where we get all of our water, it's the water supply for 750,000 people, is highly eutrophied. Okay? It's got too much phytoplankton, too much algae, and in particular blue-green algae. And another area of my research that I work a lot on is microcystins, which are toxicants made by blue-green algae. So that meant that because of that, the source of drinking water was poor, and it required a lot of treatment. And one of the things they would do is pre-treat by chlorination. And the idea was the chlorination would kill a lot of the blue-green algae, the ones making these contaminants. And so they would pre-treat, and one of the things that determines how much disinfection byproduct is formed is how much precursor organic is there, how much chlorine is there, and how long it reacts. So in this case, they were using a lot of chlorine because of the quality of the water, and they were doing it starting right at where they took it out of the reservoir, and it was several kilometers. So they had a long residence time, a lot of reaction time. So they were forming a lot of disinfection byproducts. So, chlorination in water forms hypobromous acid. Why is that important? Because then it can react with these humic and fulvic acids and form these halomethanes, which are cancer causing. So here's sort of the drinking water plan, and one of the things they wanted me to do was help them design a process to remove some of these cancer-causing agents. And it was very politically charged. People were concerned. The government wanted us to come up with a solution before we told the public all these chemicals are in your drinking water. So this is what we came up with. Long story short, it relies on activated charcoal to remove these chlorinated products. Now the problem with that is it's expensive and it's time consuming. You have to do backwashes of it, and you have to replace it a lot. And so it's really an expensive issue and time consuming. And there were a lot of times when the water system was offline because of this. And they were trying to minimize. So one of the things I did was use satellite technology to come up with a model of these blooms in the lake so we could minimize when we treated and how much we treated to only when we really needed to do it. And at the same time, we wanted to maximize removal in the treatment plant. So we used our DipicFrag non-targeted analysis, and guess what I found was most of these 600 chemicals were not just chlorinated, but they're much more potent than the chlorinated compounds. So not only were there a lot of unidentified chemicals, but they were toxicologically really relevant and a problem. So we identified what all of those were. I'm not going to go through all the methodology of how to do that, but we were able to identify all those chemicals, and we published all this and you can look it all up. So lots and lots of chemicals in the drinking water. And so we came up with a way to remove them and minimize the amount that was formed. But one of the problems, I've got a slide here. So one of the questions was how these brominated compounds were forming. And I give a little bit of that here. And this is forensics, right? We're trying to figure out what's going on and where it's coming from, what's causing it so we can stop it, what effects it has. So we were doing bioassays with all these things to figure out how toxic they were. And it turns out that, here. So in this process, there's a lot of bromide in the water coming from the mountains. It comes through it comes from about 1,500 kilometers away. So it goes through a lot of cities and farmland and things. Anyway, there was a lot of bromine in the soil, and it ended up here. And when you chlorinate, then we were actually oxidizing the bromine, so then it would make hydrobromous acid, and then it would react with these organic compounds. And so we were getting chloro compounds, we were getting bromo compounds, and bromochloro compounds. So it was a function of not only the chlorination and how much and how long, or how much algae there was there, but the fact that there was this bromine there that was causing all these chemicals to be formed. They hadn't been found before because other places people had studied, like in the Netherlands, had very low bromine content, so they never saw any of these compounds. So we found all this, and then we optimized how they treat and how much they treat to minimize how much we formed, and we came up with a treatment process using activated charcoal to get them down below what was thought to be a safe level. So this is sort of the forensics that required chemistry. We had to figure out what was there, what was causing it, and how toxic it was, so there was a risk-benefit, and then engineering treatment to figure out how to solve the problem. So we solved the problem, and the government was pretty happy. So in the end, we had a library of 700 new brominated disinfection byproducts that had never been seen before. We have a library now for all those, and other people can look at them. Future work, yeah. Okay, so that's sort of using chemistry in a forensic way to figure out what's going on in a complex situation. I want to switch gears now a little bit and talk about something called a pull-down assay that we've developed for untargeted analysis of effects. So now, if you have a sample from the environment and you get an extract of that, the question would be, what effect could it have? If we run a targeted bioassay, it will only give you information about that endpoint. And in the body, there are many, many, many pathways that could be affected. So one of the things we'll do is a pull-down assay. And we can do this really two ways. Okay. Too many words here. You can read it later when you get a copy of this. But basically what we're going to do is we could either take a single protein and react it with our extract from the environment that has all these different things in it, and then we would measure by our mass spectrometry only those compounds that are binding to that protein. So that would be a specific endpoint. We would pick a critical protein to which it might bind, and then we would be determining what things in that mixture would bind to that endpoint. We pick things like, in this case, an endpoint that's related to cancer. so to carcinogenesis. So we're interested in cancer-causing agents. Now, the other way to do this would be to take a mixture of proteins. So take a whole mix of a homogenate of liver or something that has many different proteins in it, and then ask the question with a single chemical what it might bind to. So to try to determine if we knew the name of the chemical. So here's a model example of this chemical, which we know is going to be an initiator of cancer, and we can find out what proteins it binds to. And so that would allow us to say, okay, here's the mechanism of action of that model chemical. Or we could pick a different chemical and still react it with all of our different proteins down here, okay? And see what proteins it binds to. So we can do either specific or non-specific, bottom-up or top-down. We can either know the chemical, figure out what it's doing, or we can have a mixture of chemicals that we don't know what they are and see what effects it has on a particular endpoint. So that's the pull-down assays. I want to talk a little bit about environmental forensics inside. This is an example of some mutagenic azo dyes. This received a lot of press globally in the press. We just published a paper a couple months ago in the Proceedings of the National Academy of Science in the United States on liquid crystal monomers. Does anybody know what a liquid crystal monomer is and where it comes from?
Screens.
Absolutely.
I have already one, so.
Okay, who is, somebody else said it at the same time, pretty much. There you go. Good job. Yeah, and you know, there are several thousand of them, and so we've been doing a lot of work with where they end up. And one thing you can do is monitor dust, indoor dust, where people live. And so we've been looking at a lot of these chemicals, but again, it's a big number of chemicals, most of which have never been identified in the environment, most of which there are no standards for. I'm going to just come up with just one example here of dust, because this one's important because they're cancer causing. I'm not going to go through a lot of this. Bottom line is there are many chemicals in the environment. You're exposed to a lot of them. They're in your environment, they're in your blood, and they're in your food. And many of them have never been identified, never been assessed. So we just don't know what they're doing. As far as monitoring of the environment, we monitor less than 1/10 of the 1/100th of 1% of the chemicals out there. If that doesn't bother you, it should. It's just an overview of what I was telling you about, where we would do a chemical mass balance, figure out what we've identified and what we haven't, come up with the compounds, come over to our bioassay, do our bioassay directed fractionation and identification, and in this case we would be looking for binding to PPAR, which is important for carcinogenesis. And so this is sort of the whole process. This is the chemistry, here's the bioassay, and the toxicology. So we found in house dust a number of brominated flame retardants, never been identified before. We got into big trouble arguing with people in the literature because there were no standards. So without standards, without authentic standards, you can't really prove. So I was writing structures for these things. Remember, as a kid, I memorized all these fragment patterns. So I was figuring out structures. We had accurate masses for all these chemicals and retention times, but from the fragments, we had to figure out what they were. And so we did that, and here's some of the ones that we found. There were PBDEs there, but there are a number of other things there as well. Did our high resolution mass spec, did our identification, went through this, came up with a number of compounds, about 750 to 800 chemicals, and then we had to figure out what they are, which we did, and we tested them in bioassays, and we did a bioassay potency comparison, figured out which chemicals were contributing the most to that, and it turns out that they were what are called azo dyes. They're used for color, and the one that was most predominant was the ones in blue jeans. So I did another study with some of my buddies in Hong Kong, where we looked at what comes out when you wash things. And that paper made huge splash all around the world. Newspapers were calling me from everywhere with that, because these things are very, very potent carcinogens. And so they come from your clothes. When you wash them, they go into wastewater treatment plant, but they also end up in dust. And this is a big issue in risk assessment for the US EPA because of something called pica. Anybody know what pica is?
Something for the cosmetics. No, I didn't. I'm sorry.
Pica is the term given for when you eat things that are like dust or soil. And little kids are classics for doing this, right? Crawling all around the floor. And so they're exposed to these things. So a lot of the work that we do is on what's, because companies won't tell you. And then also governments, for all the chairs and everything in this room, there are rules about flame retardants that have to be in them, okay? We were talking about that the other day with the world expert on this. But the government says you have to put them in to protect from fire. But they don't all stay there, and some come out. And they end up in the dust on the floor. And we can find those. So we find them, and then we do studies about how much would end up in people if they put their hands in their mouth. And little kids are the worst, because they're crawling around, and they're also doing pica. They eat it. Whatever they find, it goes in their mouth. So anyway, so we figured out, what was there. We did a potency balance for carcinogenesis, and then determined what was in there that was causing it. And it turned out it was the blue color in blue jeans. It's the primary one. I'm going to talk about that. Validation was difficult, and there were groups around the world saying I was an idiot and stupid and wrong. And a big battle was fought out in the literature. EPA was our American Chemical Society was refusing to publish our paper because we didn't have standards, and one thing led to another. But in the end, we proved we were right and published the paper. So anyway, it's these guys right here, these azo dyes. And let's look at that. Anyway, it's really kind of a global issue now. There are about 3,000 of these, so it's difficult to monitor for all of them. But people are really concerned about them because most of them are very, very potent carcinogen. They bind to DNA. They form adducts with DNA, and then when DNA replicates, it has to pull apart. Wherever there's an adduct, it breaks. And then the body tries to repair that, and it's pretty efficient at it, but sometimes it makes mistakes, and that's what leads to cancer. One of the things that leads to cancer. So we were looking at mutagenicity for that, so we've got a bioassay to do that, because mutagenicity is the first step with the DNA to cause carcinogenesis. So again, we do the chemistry, we do the bioassay, the biochemistry to figure out what's going on. this is just a schematic diagram of sort of how we do all this by combining chemical analysis, fractionation, bioassays, mass balance of an endpoint. And this is our untargeted strategy to identify causative agents. So if we suspect someone's sick or there's a cancer node, the group in Brno at recent talks is probably one of the world's leaders. We have cancer registries, so we can look at populations and we say, oh, here's a population that has a very high rate of cancer. Then the question becomes, why? Is it an industrial source? Is it something else? And so then we go about this process of getting samples from the environment, soil. We do a lot of air sampling, water sampling, food sampling, and then we try to figure out what might be the exposure, what chemicals are to which the population is being exposed. And we do it using this pull-down system. Okay? So now I have some known endpoints of proteins, and then I take that whole mixture from the environment and see what chemicals we can get associated with that. Here's a list of our bioassays that we use. 4 different endpoints. So here's genotoxicity, here's our dioxin-like effects, here's our, I can't read what that is, oh, the small fish model. So anyway, we have a whole range of bioassays that we can apply in this process. Okay, moving right along. So I want to talk about DNA. Remember CSI, you send it to where? Mass spec or DNA analysis, right? So I'm going to talk now a little bit about DNA. Where I couldn't decide if I want to be a biologist or a chemist. So I've done some chemistry, let's do a little biology. So in my mind, the emergence of DNA as a detectable and quantifiable unit of observation in science is probably one of the single most important advances, at least in my lifetime. This has been revolutionary. We've used this in a lot of ways, one of which, this one, we were able to use a DNA technology to determine in the environment the threshold of effects of ammonia. And this research that we did resulted in the United States EPA lowering the water quality criterion for ammonia by a factor of 10. Because we were able to show that there were effects occurring ecologically at levels that were more than 10 times less than what they were requiring. So this can have really important, far-reaching implications, and the EU often follows what EPA does and vice versa, and of course Serbia does what the EU does. So it could have effects here on your ammonia. So how else do we use this? One of the studies we've been doing a lot is with the microbiome. Microbiome can be the human microbiome, so we're talking about bacteria in the gut, and it can also be an environmental microbiome. So what we're talking about is what's called eDNA, environmental DNA. I'm not going to talk about the work we've done with human biome, but we've done the biome in fish exposed to different contaminants. And I've got a whole list of papers we've done that you can look at, and so you'll find things there about microbiome. If you're interested, then I can send you papers. But as an example, here's like an environmental microbiome, not just say nitrogen cycling. This is a big issue in many countries. And specifically, we look at the sediment microbiome. We've been doing this a lot of different places. And these are all of the different processes that the microbes are modulating. So there are a bunch of them. So in the environmental microbiome, I get samples, and it could be microbial DNA. And work we did with COVID was RNA, not DNA, so everything we do with DNA, we can do with RNA. But by getting samples either from individual organisms, or from their biome, or from the environment, sediment, soil, water, we can work out pathways that are being affected by chemicals from the biome. So this is just a sort of a pathway analysis. This is the Kyoto system. Tomorrow, I'll tell you how we worked out the mechanisms of action for PFAS, which is this process. But we have these different nodes, and by looking at the differences in DNA expression, we can figure out which pathways are upregulated and which ones are downregulated. And when I know that, then I can go through and draw a whole map, and from that we can predict what the outcome will be. Okay, this is an adverse outcome pathway. And so by looking at the biome, the microbiome, biome, and the environment, we can literally map things out. And it gets pretty complicated, and then we can pull out sections of that and look and see. So here would be steroid hormone biosynthesis. It turns out for PFAS, this is pretty critical. And we figured this out because what we were finding was there was a buildup. The body was trying to respond and making more of a particular enzyme, but the pathway was blocked, so that enzyme was just building up. And that the substrate that we're going through that pathway, we're going down. So anyway, that's how we use this. It's pretty complicated, and it takes a while to learn, get your head around some of this pathway analysis. But we've written software programs that will help guide us through this process. Environmental microbiome. How do we use this? We can monitor the environment. So for instance, and I'll give you a couple examples, but here I've got some for aquatics. But I can take a water sample from a pond and tell you what lives there. And I can not only tell you what lives there, I can tell you how many of them are there. Okay? And so we can do environmental monitoring. The old way of doing this, if I wanted to know about fish in the Danube, we'd have to go out there and catch the fish with nets, and that meant killing the fish and a lot of disruption. I was doing this in the Rhine River in another part of my life when I lived in Germany. And then, so to avoid that, we were using side scanning sonar, basically fish finders, to do that. Well, now we don't even need to do that. We just take a water sample, and then I can tell you, and we use this particularly for changes, not so much absolute, but changes. This is now, we've developed this for China. This is how they're doing environmental monitoring of all of China. We've developed this for benthic invertebrates, for fish, for a lot of different things. So they just go out and get a water sample several times a year, and then they make a map of what's there. I'll show you in a minute what it looks like. And we're looking for changes. Is it getting better? Is it getting worse? Are species getting more, fewer? Biodiversity changing? We can do this in a pond system, and I can even do it for birds and tell you what birds are using that area. We've developed a program in my province. They were interested in endangered species, and particularly it's, I told you, it's a dry area, right? And we have some tree frogs. So what do tree frogs need? Trees, right? We don't have a lot of trees, and we don't have a lot of water, so it's an issue for them in dry years. And the provincial government's interested in monitoring for them. And the way we used to do it, my daughter used to like to do this, was with peat surveys. We'd go out and listen and say, I think there's some there. Oh, no, I don't hear any. But now we just take a water sample from the different water bodies, and I can tell you if they're there or not, and and how many are there. We developed this and field tested it in an area called the Experimental Lakes District in Canada. Some of you have probably heard of it. It's quite famous. You can go there and do research. So we have a controlled situation we can do. So we did that. Another one is a gift from our friends here in Europe. I don't think there's anyone here from the Netherlands. But zebra mussels. Terrible. Okay, they have what's called a Belgrade stage, a little larval stage. They can swim into pipes and then they attach, they filter feed, and they grow. What do you think happens to the water flow in the pipe? Not good. What if that is a nuclear power plant? Ouch, bad. Okay, and so they're really a problem. They get on everything. They get on boats, they get on ships. They cause a lot of problems. And our province didn't have them, doesn't have them, and don't have them, but they're in the province next door, in Manitoba. And so the concern is, if they come, we need to deal with them very quickly. So we developed and passed off to the province a protocol to just take water samples and determine if there's zebra mussels there or not. So we can do early warning, and then they can go in and do remediation to try to keep them from spreading. So that's an example where we've used this kind of technology for endangered species and for invasive species. And we also use it for monitoring community structure, for changes due to environmental damage. A little bit longer story involves this guy. So, in China, the red-crowned crane. At my house at home, over my bed, I have a tapestry of cranes, because cranes are the symbol of longevity and fidelity. So my wife and I have 2 cranes over our bed, okay? We've been married, whatever, 55 years or something. So it seems to be working. But they're they're an iconic species there, a little bit like storks are here, right? People want to protect storks. So anyway, the populations were going down. The Chinese government set aside protected areas on the coastline of China, 5,000 kilometers. 5,000 kilometers just to protect this species. But populations were still going down. So it was decided they would develop a breeding program. They would go get some of the cranes, bring them into captivity, and breed them, feed them, grow them up, and then release them into the environment. So very expensive, very time-consuming, and they were doing this. Populations still going down. The government in China said, time to call the professor. And so I asked one of my former postdocs, Miguel Mora, who was at Texas A&M University, to work with me. And so we went to China to figure this out. We looked at the microbiome of the birds they were raising. And what we found was it was very, very different from wild birds. What was happening was, in the rearing area, they had caregivers, people, sort of like at a zoo, taking care of these animals. So they were being exposed to human bacteria. In addition to that, to avoid disease, they were treating them with antibiotics. So their gut biome was a mess. They were feeding them artificial diet, food, What was happening was when they got out and were released into the wild, they couldn't digest the food that they were getting naturally, and they would die. And so it didn't matter how many they raised. So we set up a program to minimize human contact, to feed them more natural foods, and not give them antibiotics. And it worked. Then, because we put bands on them and we were putting radio transmitters on them so we know where they go and how long they stay. I use satellite transmitters. A lot of the work I do is with birds and satellite transmitters. So we can track where they go and how long they live there. So it worked. Okay, so we solved that problem, which was good. We can look at soil microbiome. We do this a lot with oil contamination sites, to try to remediate them and see whether things are getting better, things are getting worse. Okay. Sometimes we identify individual animals and sometimes not. We can do what's called a DNA meta barcoding. So it's just like barcoding on products. But we measure taxonomic units. So I can look at the DNA. I don't know what the species is. If I know the species, that adds a lot of value, a lot of information. Because if you tell me there's moose DNA in a pond, that tells me a lot about that environment. Because moose need certain things, and they won't be there if they don't have them. So there's value added to knowing the individual species, but you don't need to. You know, you can have what we call meta barcoding, and that we just identify operational taxonomic units, OTUs. I don't know it's a human, I don't know it's a moose, I just know it's a species. It's not as good as targeted, but this is an untargeted way to monitor the environment. So some of the advantages are it's rapid, it's cost effective, it's more comprehensive, because if again, you find what you look for, right? If we're not looking for anything in particular, we get everything. And we can monitor biodiversity, and we can monitor for invasive species. We don't necessarily need to know what they are. So we've done a lot of this. Xiaowei, one of my former students, is now the director of environmental monitoring for all of China. And they have adopted eDNA monitoring for the whole of China. I'm not going to say too much about that. So here's sort of what we find. This is an example of Taihu. Hu in Chinese means lake. Anybody know what Tai means? Sometimes pronounced Tai. Same. It's the same in Japanese. The same kanji character. Dai. Big. Right. All right. Good job. It means big. Anyway, Taihu is a very, very, very important lake in China. It's iconic. People like to go there, and it's really degraded. The ecology is really in trouble from lots of things: industrial inputs, wastewater, all kinds of things. And it's so important that the Chinese government said we need to do something. Now, I'm an Einstein professor of the Chinese Academy of Science. That's their version of the Nobel Prize. And so again, they have a problem. Who do they call? They call me. Professor, come over. We need to do some studies. What are we going to do? And I said, hey, why don't we apply the DNA technology and see what's going on? So I'm not going to go into a lot of the detail. Anyway, where was it? I thought I had the paper in here. Anyway, there are a couple of papers on this. So we were looking at the Jiangqiang, which is you call Yangtze River, and it's got a lot of lakes and rivers and things, a part of it. And we were able to measure DNA. So we took all the important species and we did sequence their DNA for all. So this is things we know are there. This is not meta-barcoding. This is identifying individual species. So we have all those here. We have all these different locations, and we can quantify this. So we've got a whole library. You know, I'm big on libraries. And this is the relationships for all of these animals. We got fish, we got zooplankton, phytoplankton, every species that we know that occurs in this lake. And now we can monitor status and trends just using a little bit of DNA. So we take a sample, we extract the DNA from everything as a group, and then we use polymerase chain reaction to amplify the DNA, and then we use barcode sorting and ultimately identify all the individual species and quantify how much DNA is there. I don't like to use absolute amounts of DNA, I like to use relative amounts. So it's better for me to say the relative proportion of these species is changing as opposed to the absolute numbers. Okay? You can do absolute things, but it's for monitoring like this, it's really better to do. So we've got our database of all the different species. There's some more. This is the zooplankton from Thai Lake. All the different species. About 40 species. We can then start to look at trends of interspecies and intraspecies divergence. This is over time, how things are changing, whether things are getting better, getting worse, what biodiversity, is it going up, is it going down, and we can do all of that. Here's some of the macro invertebrates that are important there. Again, we've mapped out all these. We have the DNA sequence of all of these animals in our library. These are all the fish species, very important. Can you read those names? So, this is traditional kanji characters. So then we do this for a whole local community. So we have everything that's going into the lakes. We can map out biodiversity. So this was in 2014. We can map changes in the fish community. So we take samples all around the lake, and then we can map that out and look at it over time. See if it's changing. Is it because they're doing a lot of remediation around the lake? It has a eutrophication problem. They're trying to reduce phosphorus inputs. It has industrial inputs. They've stopped. And our job was to sort of see, are these helping or not? And because they're very, very expensive to do, to upgrade all the wastewater treatment plants and everything. It takes a huge amount of money, time, effort. And so we want to see if it's having an impact. They're planting macrophytes all around the shoreline to minimize things coming in. So it's a huge, huge effort, still ongoing. So we're continuing to monitor them. This is where we worked out the effect on unionized ammonia. Long story short, we were able to look at, measured how much ammonia was there, total ammonia and unionized ammonia, and look at what species diversity was occurring. And we were able to come up with a threshold value that was affecting diversity. And that then when we published the paper on this, the US EPA called me, and then we started talking, and one thing led to another. A number of my former students worked at the US Geological Survey Lab in Columbia, Missouri. They started doing studies, and the result was they lowered the standard. But it all came from this paper. We can compare things among locations and over time and see what the trends are. This is just sort of overall where things are going. A lot of this is being done in Canada now, United States. U.S. Geological Surveys hired a couple of my former students, Kathy Richter, for instance, to run their program in environmental monitoring using ED&E. And what was that paper? Okay, just sort of one last thing. Are we okay? So, I'm an advisor to a lot of governments. In Canada, I am an advisor to the Minister of the Environment, and in the EPA, lots of, and provincial. And in Canada, one of our big issues is something called oil sands. Anybody heard of oil sands? So it really disturbs the environment a lot. It's oil associated with sand, so they have to dig up all the sand and then extract the petroleum hydrocarbons, and then there's a lot of waste water associated with that. So this is what it looks like. This is the Athabasca River. This is in the north. This is an industrial complex, and over here are some of the mining areas. But the result of the wastewater is not allowed to be released to the environment. It's put into ponds, waste ponds. And the idea is that they should be able to get remediation by sitting there, and they're not really improving much. So if you take samples, they cause acute toxicity, they kill things, cause deformities, and all kinds of effects. They're about, our best estimate is there are about 200,000 chemicals. 200,000 chemicals. So, what do we do to try to regulate this? The government came to me and said, John, we think you're a pretty smart guy. We want you to give us a model to predict these effects before we release any of this water into the environment. So we did a whole bunch of bioassays. Some more here. We did our pull-down kind of assay, a whole bunch of things. I'm not going to spend much time on that because we're sort of running out of time, I think. We'll talk about this another day, I guess. So, what we did was figure out the mode of action, and it turns out most of these chemicals are what are referred to as naphthenic acids. So they're water soluble, but they have a wide range of structures. Some have heteroatoms, oxygen, sulfur, nitrogen. Anyway, I was able to work out the critical mechanism of action. So what is the critical mechanism of action? The critical mechanism of action is the effect that occurs at the least concentration. So it's the one that you can have other things happening, but it takes a lot more of it before those happen. So we worked that out, and I was able to come up with a model that predicted toxicity to a range of aquatic animals based on their structure. I'm sorry, I don't want to talk about that. I thought I had another slide in here. Too much detail for you, sorry. So this sort of shows what I think I wanted to show. But here are some pictures of the range. This is of different extracts. So this is our bioassay directed fractionation, and we were able to figure out where most of it was, and it turns out the mode of action for most of these chemicals is something called narcosis. Narcosis is what they give you for an anesthesia if they give you gas when you're having an operation. and it's a reversible process. It goes into your nerves, blocks them, they swell, it blocks them, but then when you breathe it out, you recover. But if you don't breathe it out, like the fish can't stop breathing, they die. Anyway, we were able to do this. I was able to develop a model that predicted that based on the molecular volume of each of the chemicals. And then we came up with a model based on our mass spectrometry. I named all the chemicals. I didn't know their structure. I just gave them a number basically. But I knew their mass. I knew their accurate mass. And I was able to come up with a model that was about 95% effective in predicting toxicity based on the untargeted mass spectrometry and the untargeted toxicology. I know that's a lot to try to get your head around in a short period of time, but the bottom line was it worked, and it meant then that the government could start releasing some of this to the environment. Because I was telling them, if you don't, you're going to have a disaster, because these ponds are right next to the river, and they're eventually going to fail, and they're going to break out, and when they do, it's going to kill everything all the way down the river, which would have been a disaster. So we worked out how much we could release over a slow period of time and monitor with our model and our untargeted chemistry. And so we're bringing that down. We're getting a really bad situation improved. So I'm not going to go through all this again. We'll stop there. If you have more questions about what we do or how we do it, I'm here tomorrow too. But anyway, thank you for letting me come.